known as Meningeal worm, deer worm, or brainworm. It is a parasite common to white-tailed deer, which causes damage to the central nervous system. Moose, elk, caribou, mule deer, and others are also susceptible to the parasite, but they are what's called a dead-end host and are infected in neurological ways instead of the meningeal tissue. The frequency of infection in these species increases dramatically when their ranges overlap high densities of white-tailed deer. The life cycle begins in infected meningeal tissues in the central nervous system, where adult brainworms lay eggs. The eggs are dislodged from the central nervous system and passed into the lungs, where they hatch. The larvae are then coughed up, swallowed, and proceed through the gastrointestinal tract. Snails and slugs then serve as intermediate hosts, which are later eaten by their true host, allowing the process to continue and cycle all over again. Changes in climate and habitat beginning in the early 1900s have expanded range overlap between white-tailed deer and moose, increasing the frequency of infection within the moose population. Life cycle. The life cycle of brainworm is complex and multi-staged. Adults lay eggs on the outer layer of the brain or directly into the bloodstream of an infected host. The eggs hatch into first-stage larvae, which travel in the bloodstream to the lungs, where they travel up the respiratory tract, are swallowed, and then pass out of the body in the mucus coating of fecal pellets. Gastropods, such as snails and slugs, feed upon the mucus coating of the fecal pellets and ingest the larvae. While in the gastropod, the larvae develop into second and third stage larvae, which are capable of infection. Gastropods, carrying second and third stage larvae, may be accidentally ingested with plants, which results in the larvae being transmitted to the new host. Larvae then move into the new host's stomach wall and make their way into the central nervous system, as they do in the white-tailed deer, or the brain in other mammals. Once in these tissues, they develop into their adult third stage of life and lay eggs to begin the cycle again. Transmission. Adult brainworms can persist for many years in a single host, which allows for many first-stage larvae to be shed in feces. It is quite common in many populations of white-tailed deer which have built up a strong resistance. After gastropod ingestion, moose or other deer may be hosts to the second and third stage worms. Moose resistance to brainworm is much lower than white-tailed deer, which results in a higher mortality rate. Infected deer density, temperature, climate conditions, and length of transmission periods all affect transmission levels. Areas with higher deer populations experience dramatic increases in incidental brainworm larvae consumption. The temperature and climate conditions of summers can regulate gastropod abundance, with hot, dry summers reducing populations and cool, dry summers increasing populations. High or low gastropod populations directly affect transmission rate of larval worms to new hosts. Hot, dry summers also reduce the survival numbers of first-stage larvae on fecal pellets. Variation of transmission periods is based on the time when snow is absent from the ground. Gastropods are dormant during snow periods, thus first-stage worms cannot be transmitted to additional hosts. So, shorter seasons of snow are met with longer transmission periods of increased potential for transmission through the intermediate vector gastropods. Brainworm affects neurological and behavioral responses. Deer rarely show any external symptoms. of brainworm infection due to their high acquired resistance. Moose, however, have low resistance and may show a number of symptoms. Though infrequent, cases of moose recovering from brainworm infection have been reported. In both deer and moose, symptom severity does not necessarily vary with severity of infection. Infected individuals may not have any external symptoms. Mild symptoms may include slower movements, slower response time, frequent stumbling, unusual tilted head, and emasculation. Severe symptoms include extreme weakness, lameness, walking in circles, partial or whole blindness, loss of fear of humans, and mortality. Several other creatures are susceptible to brainworm infection, including elk, caribou, mule deer, sheep, goats, alpaca, rarely cattle, and rarely horses. Severe neurological damage, similar to that of infected moose, is shown to occur in these species as well. Diagnosis. Presently, a commercial antibody test that can detect brainworm before death does not exist. Diagnosis in deer can be conducted by analyzing fecal pellets for larvae or post-mortem autopsy to detect presence of adult brainworm in the brain cavity or second and third stage worms along the spinal cord. However, brainworm larvae are difficult to distinguish from other parasitic worm species that can also be found in fecal pellets, so detection of adult worms through autopsy is recommended. Diagnosis in moose is conducted with autopsy to detect worms in the brain or spinal cord. Diagnosis in horses can be conducted with post-mortem samples. History. In 1912, an unknown neurological disease affecting moose was first reported in Minnesota. Major declines in the moose population were reported. 1925 to 1927 and 1933 to 34, following the discovery of this disease. In 1963, a Meningeal brainworm was determined as the agent causing neurological disease in moose. White-tailed deer populations in the eastern United States are now known to be commonly infected with the Meningeal worm. Infection in the southeastern and western United States is less common. This disease is also found in the Canadian provinces of Ontario, Saskatchewan, Manitoba, and Nova Scotia. The grassland biomes of the United States and Canada apparently act as a barrier to the movement of the disease. Evidence from Canada seems to support this, since the western distribution of Meningeal worm has changed little since the 1960s. Areas. The geographical ranges of moose and white-tailed deer were historically separate prior to the 20th century. Moose are well adapted to winter survival, whereas deer are not. They could not withstand the harsh winters in these regions of the northeastern United States and southeastern provinces of Canada. Deer populations began to move into the southern portions of the Moose Range in early 1900s following changes in climate. Logging, mining, forest fires, and increased human development all contributed. High-quality deer habitat associated with these changes has led to dramatic increases in deer abundance in these regions. White-tailed deer are the normal host to brainworm parasite and have adapted to its presence. Deer and brainworm have co-adapted in an evolutionary arms race over time. Deer remain largely unaffected by the presence of brainworm because of this immunity they have built as a result of this co-adaptation. The prevalence and infection rate of brainworm in deer is density dependent. Increased rates of infection by the parasite are the result of higher deer densities. Moose populations on the southern fringes of their range have recently experienced dramatic declines. As deer encroached upon the southern fringes of Moose Range, they introduced the parasite to a new host, the moose. Upon transmission of the pathogen into the moose, the worm causes cerebrospinal disease, a disease of the nervous system that also results in death. Brainworm may be one factor that has contributed to moose declines in the southern portions of their range. This distribution of host pathogen dynamics due to habitat alterations has promoted the spread of this parasite to this new host and has potentially contributed to the declines in moose abundance and productivity in regions with high deer densities. Continued increase in deer abundance and densities in the southern fringes of Moose Range are predicted to facilitate sustained transmission of brainworm to the moose. Continued transmission of deer-related parasites, coupled with low productivity and habitat degradation, leads to a troubling prognosis for southern moose populations. It is predicted, if the current trend continues, moose populations in these regions will not be viable and population decline will persist. Treatment. Currently, there is no definitive treatment for brainworm in mammals, though research is still being conducted. The use of ivermectin, as well as other similar medications, have been attempted in white-tailed deer. The results indicate, however, that ivermectin was ineffective against larvae that had already reached the spinal cord. Ivermectin combined with anti-inflammatory therapy have been used to manage infections in goats. Current and past research. The majority of moose brainworm research in North America has been conducted in northern Minnesota, where a historical moose population of 4,000 to 5,000 moose had declined to an estimated 1,200 individuals by 1997. In this region, wildlife managers are challenged to predict and attempt to mitigate moose declines resulting from deer-related pathogens. Because no effective method to prevent transmission or infection of brainworm in moose have been found, managers have focused their efforts on decreasing deer densities in these regions. The Minnesota DNR identified that population densities greater than 12 deer per square mile result in increased moose mortality as a result of more brainworm. Managers in these areas are responsible for evaluating suitable habitat for moose and deer, as well as setting management priorities and population goals to decrease the transmission of brainworm to moose. Though the main focus of research has been on white-tailed deer and moose, research has found that the guinea pig can be used as an experimental model of brainworm infection.